Hi, good afternoon. My name is Chanel Hasten. I'm the Director of Outreach and Community Relations for the Elakha Alliance. And if you're unfamiliar with our organization, I will give you a quick little summary in about 1 minute. Tonight, we have a presentation on sunflower sea star recovery with Ashley Kidd, which we're very, very excited to hear from today. So I'm just going to start off a quick little introduction about the Elakha Alliance. So we are a nonprofit based in Oregon. I'm sorry, somebody just started some yard work I don't know if you can hear that. If you can't, then that's great. If not, I will go close the window behind me, or my assistant will do that. Perfect. Okay, apologize about that. So the Elakha Alliance is a nonprofit based here in Oregon, working to return sea otters to the Oregon coast. We were officially a nonprofit about 5 years ago. We were started by the late David Hatch, who's a Siletz tribal member, as you can see here in this photograph. The photo on the right there is the most recent picture of our board. We just met the other week in Coos Bay in Oregon. There's a lot more information about us on our website, but that's just a quick little summary about who we are. As I mentioned, our mission is to restore healthy populations of sea otters here on the Oregon coast, and in the process, help make our kelp forests more robust and resilient, which benefit everyone. So ultimately, what we want is an ecosystem in balance, and that is with two key predators in the kelp forest and estuary ecosystem. The sunflower sea star is one, which we'll talk about today, and obviously the sea otter. But unfortunately, we've had a really big shift in our ecosystem due to a lot of different factors, which I'll talk about right now. So on the West Coast, there was maritime fur trade, which took out an estimated 150,000 to 300,000 sea otters. That was the current population, and pretty much made them locally extinct across the entire Western Pacific. About 99% of the entire population was killed for their fur, unfortunately. So that is what happened in Oregon. They became locally extinct. So we are the only state in the Western Pacific to not have sea otters on our coastline. And then sea star wasting disease happened in 2013 through 2015, and I'll let Ashley cover that in a couple of moments more in depth. So ultimately, this is kind of a sneak peek of what our current kelp forest ecosystems look like on the West Coast. And I think Ashley will also talk about this too, so I'll let her give the shining light on that. But this is what we can agree. We all want a really healthy kelp forest ecosystem that benefits all the organisms that depend on it, and that is with sea otters back in the Oregon ecosystem. So how you can help the Elakha Alliance, there's several ways. You can donate, become a monthly donor for as little as $5 a month. Is super helpful to us. You can go to OregonSeaOtters.org, is a little easier to remember than ElakhaAlliance.org. You can also sign up for our RAF newsletter that goes out once a month from me, so you can stay in tune with what's happening here in Oregon, and also just spread the word that sea otters are really, really important keystone species in our kelp forests and should be back there. Also, follow us on social media if you like cool, cute pictures of sea otters and also scientific information that is helpful and knowledgeable. Follow us on the socials. And last but not least, I'd like to welcome our guest presenter, Ashley Kidd. She's the Conservation Aquaculture Program Manager at the Sunflower Star Lab in Monterey Bay in California. With 12 years of experience as a professional aquarist and public aquaria, she draws upon her industry knowledge of animal husbandry and welfare, marine invertebrate conservation, and scientific diving to contribute to part of the roadmap for sunflower star restoration pertaining to conservation aquaculture. Woo! That is a great introduction, so I'm going to pass it off to Ashley. And also, if you have any questions, please feel free to use the Q&A feature on Zoom or type it in the chat, and we'll get to it after she's done. Thank you.
Hi, everyone. Just taking a few moments. Can everyone see the title slide?
Yes.
Awesome. Okay. Well, thank you, everyone. And thank you, Elakha Alliance, for inviting me to talk about one of the other keystone species of the kelp forest ecosystem. A little background about the Sunflower Star Laboratory. We started in 2021, and we represent a community of motivated citizens in Central California that want to support the resilience of kelp forests through species restoration. So we are also a nonprofit organization, and we are now 2 years, or not even quite 2 years in, and it's been an incredible ride, and it could not have been done without the collaboration and the partnerships that have been built before we began as a lab and since. So hopefully I'll be able to give you a little overview of all the momentum that we've had and all the work that is still yet to be done. So I hope to cover what is the sea star. I'm sure there are many people in the crowd that know about sunflower sea stars, and we all loved seeing them underwater or working with them. I had the opportunity to work with them while I worked at the aquarium in the Pacific, and hopefully shed some light on some cool facts, because I've got to, compete with these super cute sea otters, and then go through all the things that have happened to them in the last decade and what we're trying to do in the next decade. So Pycnopodia helianthoides is the scientific name for the sunflower sea star, and I like to talk about it particularly because it actually has a name that makes sense when you translate it. Pycnopodia just means dense feet or many feet, and helianthoides is like a sunflower. And I thought they got that name pretty accurately. This particular species is a type of sunflower sea star that has extra rows of tube feet. So they have almost 15,000 tube feet that help them move swiftly around throughout their environment. Sunflower sea stars are related to sea urchins. They all are in this phylum Echinodermata, along with sea cucumbers, crinoids, all the other bat stars and sea stars, and then brittle stars and basket stars and all the fun stars that you can see in the Pacific Northwest. Yeah, basket stars are pretty cool. The Pycnopodia helianthoides is the largest known member of the class of true sea stars in size. There are ones that are heavier. They have still more skeletal structure. They're invertebrates, but they still have calcium carbonate skeletons that help support their structure. But these guys are very fluffy. They look fluffy, and they also don't have lots of calcium carbonate skeleton to support them. So there are heavier ones, and there are some longer armed ones, but they're pretty big, and they're definitely the ones that if you were coming across them diving, they would be the biggest ones by far. So this is sort of generic sea star fun facts, which I think are really cool. And if you don't know anything about sea stars, you can hopefully share this at your next dinner party on cool sunflower or sea star facts. But in the top right, a lot of people don't really think about how sea stars eat. Sea stars can actually avert their stomach, so the two little clear pouches are actually the sea star putting their stomach on the outside, and they actually digest their prey on the outside and then unceremoniously slurp up their food once they've digested it. So they can eat pretty much anything that they can get over, and size isn't so much the matter. It's, for clams and mussels, they need to like pull it apart, but for urchins, they can kind of just digest all the tissue and everything on the outside and then get into the inside. So keep that in mind for later. In the middle, this is a juvenile sea star, but I chose it because those red dots are actually primitive eyes. So every sea star that you come across has the same organs in each of those arms. And that also means that they have an eye on the tip of each arm. So they can see light and dark. They're primitive eyes, and they're not, they don't have a cornea and a retina like we do. But they can see light and dark at the tip of every arm. So Pycnopodia that can have up to 24 arms, they can have up to 24 eyes, which is a fun fact. And then if you look on the left, they have all those amazing tube feet that all use water for jet propulsion. They can suck onto a surface with the suction cups, and then they can release it. And they basically use a combination of chemosensory or chemical sense in the water and the light dark to kind of move around. And there's no brain that's controlling every individual foot or tube feet. They just sort of all happen to move. If one releases, if one connects, the other releases, and they just kind of, there's no front or back to a sea star either. Every way, whichever way it's moving in is the front. So just fun things to ponder when we delve into the world of marine invertebrates. And I won't go into the incredible life history of sea stars, but just take note that they don't just show up in their little lives as sea stars. They have a whole pelagic stage, or several pelagic stages, before they even turn into something that looks like the sea stars that we all know and love, the 5-armed sea stars. So there are males and females with this particular species of adults. You can't tell them apart from the outside. They look the same, but they will spawn based on environmental cues, which are still being discovered on exactly what it is that triggers them. And then they have external fertilization. And as you go around this clockwise, you'll see the egg developing into 2 different larval stages. So they go through 2 metamorphoses essentially. And then this brachiolaria has this little rudiment that turns into a very orange. You can kind of see the picture in the corner that Jason Hoden at the University of Washington took of a larvae with those big long arms settling on a little piece of coralline algae, and it's suctioned onto the coralline algae, and then what's going to emerge as the little 5-armed sea star that we know is that little tuft at the top that kind of looks like the head, of which it's not. It's just what will become the sea star. So their larval stages are really fascinating. There's lots of work to be understood onto where they like to settle and all of that. And we can discuss that as much as you like, moving on. And then just to note that they do all start off as 5-armed sea stars. And then they slowly will add more arms as they go. And it's not always exactly 24 arms. It can be up to 24 arms. All right. So now that we know everything there is to know about sea stars to make you fans for life, and just mesmerized by them, now where do we find these sunflower sea stars? So as we're talking in the Elakha Alliance, they were found in Oregon as well as California, but really all the way from Baja, Mexico, up to the Aleutian Islands in Alaska, which you can see on this map on the slide. And they could be found in kelp forests or in eelgrass beds, as the previous picture showed, as well as just mud flats or artificial environments. You could find them in pier pilings, and really deep down to 400 meters. Although generally you find them from the just subtidal tidally 0 meters to about 30 meters is actually where they're most commonly found. And they do like to live between 4 degrees to 14 centigrade, which is like the 50s range. But actually in Southern California, some have existed in regions where the temperatures reach up to 18 degrees centigrade. So they do actually are found in pretty wide-ranging environments. But the sad thing is currently we don't find these sea stars in Oregon, California, and Washington. And the reason for that was still a huge area of research that is not my area of expertise, but the sunflower sea star syndrome, or the sea star wasting syndrome, that started in 2013 and turned into an epidemic that lasted 5 years, started in Washington, spread down to California, and then made its way up to Alaska. And the results are shown in this map where it's showing the percent decline. So southern Washington, Oregon, California, and Baja lost all of their sea stars, so they're now functionally extinct, similar to the sea otters that are in Oregon. So Oregon's now battling the fact that they have 2 keystone predators that are not present in their marine ecosystems. There is a group of researchers that part of the Pycnopodia Recovery Working Group, which is a large consortium of researchers and citizen scientists and nonprofit and aquarium institutions from Mexico, Canada, and the US that came together and petitioned for the sunflower sea star to be listed as critically endangered under the IUCN. And recently, the sunflower sea star has been petitioned to be listed under the United States Endangered Species Act, and it has to go through Congress to list it officially, and that will happen in March of 2024. So the listing is still pending, but it was recommended that it was listed as threatened across the whole US territories and states. So what happens when a sunflower sea star disappears from an ecosystem? As we saw in a previous slide, we go from these really dense kelp forests with high biodiversity to the urchin barrens, which is now unfortunately not an unfamiliar sight to divers, snorkelers, fishermen along Oregon and California. And this is, again, just an imbalance in the ecosystem players. And so how that broke down between Oregon and Northern California, Central California and Southern California are that, yes, we lost the otters due to the harvest of these animals for their pelts. And Monterey in Central California has had a successful recovery program through the Monterey Bay Aquarium. And so they have these sea otters that have come back in certain areas. And in Southern California, they exist off San Nicolas Island in one very remote area. So not really everywhere in Southern California, there are sea otters. But then we had the wasting disease and all the sunflower sea stars disappeared. Both of these animals eat urchins. And in Southern California, so they still had, they have a different ecosystem. They have warmer waters, different different fish and invertebrates that also eat urchins. So in Southern California, even though we lost the sunflower sea stars, we have the sheep's head wrasse and the California spiny lobster that eat urchins, whereas in Central and Northern California and Oregon, these animals don't exist, so they can't make up for that loss of predator. So unfortunately, Northern California and Oregon, they've lost both of their sea otters and sunflower sea stars. And this is a great slide that you can, delve into in the recording. And it's just a really interesting study in Central California that shows why having functional resiliency by having multiple predators enacting their predation or just their presence of a predator in an ecosystem can have profound effects. So in Central California, in Monterey, there were areas where there are sea otters and areas where there weren't. And so the study by John Smith from 2021 just shows that when the sunflower star disappeared, the urchin population increased, but the sea otters, through long-term monitoring studies at the Monterey Bay Aquarium, they saw that the sea otters actually started eating more urchins as a response to their environment. There were just more urchins coming around. So unfortunately, they don't want to eat the urchins that occur in barrens because, as you may or may not be aware, sea otters need to eat a lot of food because they don't have blubber. And so they need to rely on their food to stay warm. So they started eating the purple urchins that were in the kelp forest. So the ones that still had all that nice, yummy food and had, nice gonads that they can eat, that was great. So they were able to keep the patchy kelp forests in California, in Central California, but the urchin barrens sort of were able to stay because the sea otters weren't interested in eating those. So how can we help restore the balance? So the same group of people, the Pycnopodia Recovery Working Group that put in for the IUCN critically endangered status, then through the guidance with the Nature Conservancy, were able to put together a roadmap to recovery for the sunflower sea star, identify objectives and action items that we needed to answer in order to make a insightful and strategic recovery plan. And this really did take a village of people from public aquariums, from nonprofits, from conservation planners, disease ecologists looking into the sea star wasting, but also aquaculture professionals that would actually look into how would we recover the sea star if they are functionally extinct and extirpated from an area. What's the likelihood of them returning to an area where they're functionally extinct? How long is that going to take? Can we afford to wait for natural recruitment? And how can we help facilitate or accelerate that recovery? So what do we know? That the epidemic is over. Sadly, that took 5.6 billion sea stars with them. And the wasting is still occurring, but patchy. And there's still no smoking gun or silver bullet that would explain what exactly triggered and caused this spread, not just with the sunflower sea stars, but with the 20 other species that were identified to have a wasting symptom. So again, now the question is, just because they waste away, what exactly is causing it? If it is just stress, there seems to be many stressors that can cause this symptom. the heat wave that occurred in 2013 that definitely exacerbated and accelerated the wasting event is one major thing. But density dependence wasn't an issue. There were high and low density populations of sea stars that wasted. Some were in contact with sea stars, some were not. So it's still a really big question mark on, even though there's been great progress, there's still no clear answer. Also in the last 4 years, captive rearing has been shown to be possible through the Friday Harbor Labs. Jason Hoden and his team has been researching all the ways in which we can captively rear them, but not in a scalable environment. So in a scale that would be required for recovery across an entire continent. And even though observations of Pycnopodia have been recorded, we need to know, it would be great to get those observations into genetic samples to understand sort of how they're coming back, which is actually one of the major research goals, and the fact that urchin barrens still persist even with all the great work in Central and Northern California to understand how removal of urchins help, but there's no sustainable way to keep the urchins from coming back naturally. So, so specifically, when I joined this conversation, I came from an aquarium background, and I had worked with sunflower sea stars. And if we didn't have animals to work with in the wild, and even if we had, if we were able to collect them, there's a lot of questions in terms of biosecurity and the health of the sea stars, and should we be taking them out of the wild? So I turned to the populations that have been held in public aquarium for decades in some instances. So there's a summary of currently all the institutions, whether they're Association of Zoo and Aquarium, public aquarium, or research institution, or an aquarium accredited with someone else, and where they are and where the animals were sourced from. So in the last 2 years, because of all the work that's been done with the roadmap to recovery, and because of all the work that's happened in terms of talking to the aquarium staff on the husbandry and observing spawning events and identifying which animals are male or female, we've been able to get the species listed as a safe species, which is an AZA, or Association of Zoos and Aquarium, program that then links these few aquariums into the entire community of zoo and aquariums in North America. So it really pulls in, it allows messaging and outreach to occur and support to occur from all the different AZA institutions and share resources for this species. So that's been really wonderful. And specifically for California and Oregon, it's great to know that we have animals that have been spawning. So this little graph is just showing the information. The wild animals up at the top are from Alaska and BC from researchers, and from Friday Harbor Labs spattered in there, but all the other data points on this graph are just from the last 2 years, and public aquarium staff taking note, looking under a microscope, and figuring out what they have, males or females, when they spawn. Because prior to this, no one was able to see when a sea star spawned in the wild. It's just hard to tell. We don't understand what causes it. If there's a lunar cycle, we're looking now at when these events occur, if it has any affiliation with the lunar cycle. But we're actually noting that, yes, there seems to be a strong spawning season from January to June, but we're seeing spawnings pretty much every month of the year, which prior to that, this year we didn't know. And the latitude just helps show perhaps any discrepancies in spawning based on locality. And what we've also known just from observation in aquariums is that the same individual will spawn more than once a year. And we didn't know that for males. We were, again, Friday Harbor Labs up in Washington, who's had captive animals for their culture research. They've noted them spawning and the male spawning, but now we have, multiple institutions reporting that the same female spawned, a month or two apart. So this is great information to know when you're trying to build up a recovery plan and what's possible and what's not possible. And just so you can see, this is actually a video. Oh, there we go. Video from the Vancouver Aquarium showing us from the visitors station the spawning event. So there's a lot of flow in the exhibit, so it's getting blown away really quickly, but it kind of looks like dry ice kind of coming off of the sea star. And I can't recall whether they identified whether it was a male or female. Again, it's hard to tell when you're far away through the glass. And yeah, but eggs normally sink, but it's hard to tell with the with this particular video. But it's been great just to have the engagement of the staff and the excitement shared with the visitors. So where have marine invertebrates, or where has there been marine invertebrate recovery efforts? Coral restoration is a global investment, and looking at all the work that different institutions and organizations have put into marine invertebrate recovery efforts with their unique challenges, their interesting life cycles. We turn to their successes and lessons learned in order to build a better next generation version of marine invertebrate recovery efforts. So even in the Florida Keys, or in Florida, the Florida Reef Track project, if you didn't hear, this summer was not a great year for corals in Florida, and they're also trying to bring back urchins in a funny comparison, because coral ecosystems do best when there isn't an overgrowth of algae. And the urchins in the Caribbean went through a wasting event in the 80s, and they thought, oh, we'll give it time and maybe they'll come back. And it's been 40 years and they haven't come back. So there's been now an effort to restore urchins into the Caribbean. So we've been looking at their ways of rearing and restoring them, and discussing best ways forward through their partnerships and collaboration. And specifically in California, there is a precedent to restore marine invertebrates. The white abalone was the first mollusk listed on the ESA back in 2000. And they really did have to jump through hoops, being the first marine invertebrate listed on the Endangered Species Act, or mollusk on the Endangered Species Act. And that's where I learned a lot of the restoration strategies and met all the diverse partnerships that really brought all these different pieces together. And so you kind of start seeing the themes of, they have to captively spawn. You have to understand the habitat use and the needs, outplanting methods, and long-term monitoring. Now, the funny thing with sea stars is that, with a marine mollusk or a coral, you can put a nice little tag on their shells. And I don't have a picture of this, but you can just take little super glue and stick a nice little tag and track all your animals and see where they go and how long they live. And with corals, you can do the same thing. If you outplant them onto a reef and you're gluing them onto a rock with epoxy, you can put a little tag to track them that way as well. But with the sea stars don't even, they can eject anything that you put into their bodies, whether it's a stainless steel ring, whether it's a tag, whether if it's an implanted tag, they can just get rid of it. Nothing stays on. They've tried tattooing sea stars as well. So really monitoring these sea stars in the future will be a big area of how we want to go about that. But before I get ahead of myself, the great thing with the partnerships is that because of the work that Jason Hoden did at the Friday Harbor Labs and the need identified in the roadmap to expand the number of partners that can raise these animals, because we need just more people that have the capacity to raise these animals. We actually were able to send sperm from Friday Harbor Labs to Omaha's Henry Dorley Zoo that has a female California sea star. And these are pictures from Ben Morrow, who's one of the senior aquarists there and does the temperate aquaculture, and his first successful attempt at raising sunflower sea stars. So just this year, we were able to at least prove that Jason's work can be replicated, and we were able to do it in Omaha because Omaha doesn't have any restrictions on moving an animal, a marine invertebrate that has a potentially lethal effect on other invertebrates or other stars, because they're in the middle of the country and there's very little risk. So that was a great partnership, just to slowly make these little proving grounds in advance, and gain some confidence and more knowledge sharing all that we learned. So on top of being able to show that we can replicate the rearing that Jason's done and build upon it, we're also trying to figure out how can we work with such few animals when we don't know what triggers them to spawn, and we have a very few scattered all throughout the West Coast, but also some isolated areas. And we want to make sure that in the future and down the road, we have as much genetic material to work with to have a good, robust restoration strategy. So another very cool partnership that we started a year ago, actually, last November, was a partnership with the San Diego Zoo and Wildlife Alliance. So looking outside the box, not to our aquarium partners or to our academic partners that are all big invertebrate nerds, we reached out to the cryopreservation and reproductive team who work primarily on mammals. And they had such a blast coming up to Friday Harbor to attempt cryopreservation protocols that were developed by coral restoration, cryobiologists for corals and urchins, and working with them and discussing sort of the unique challenges or how to set up a good set of tests to know which cryoprotectants work best and what freezing rates work best. I learned all of this in the last year, and I'm by no means an expert, but it's just been really amazing to be able to bring all these unique skill sets from all these different, somewhat disparate places and actually have a successful cryopreservation protocol at the end of it. So one of the big milestones we got this year was just being able to show that we can successfully from one actually two males sperm, we were able to successfully cryopreserve them. So it's really just set us up for proper trials to show that we can do this with, you know, a dozen individuals and all produce viable offspring. And this is great for just long-term species diversity. We can now go around and with the ones that we currently have in at different institutions, we have all of the genetics being run by Lauren Scheibelhut, who was from the University of Merced, who's leading all the genetics for sunflower sea star recovery. And so with the work of the genetics coupled with the cryopreservation and our aquaculture division, we can hopefully have the right tools to make the best decisions when we are talking about recovering the species. We're not there yet. What we don't know, well, we still don't know exactly what the etiology of the disease is. We're currently working on answering questions about aquaculture, reintroduction, and monitoring. These are all the next steps. So scaling up all the work that's been done. Are there differences, you know, that when you start culturing en masse, and how are we going to go about it? So you guys can help. Whether you're a student, a biologist with training, or a biologist from the community, iNaturalist, and the Marine Agency Rocky Intertidal Network are wonderful platforms for anyone to log sea stars. And the way that you help our understanding of recovery is that if you see a sea star of any type, whether it looks healthy or not healthy, these observations will help everyone understand the spread or return or hotspots for any wasting events that we see. And because the wasting that we see is able to transfer amongst species, we want to know where that is and sort of continue to observe the way that this sea star wasting syndrome is persisting and how that might affect recovery. And if you want to know more about how to identify wasting, the Marine Network has a whole section on their website on how to identify it, how to know more and learn more about it. Again, sea star wasting syndrome affected many species, so there's a whole working group on that, not as well as people that are specifically looking at the sunflower sea star because they were the most severely affected sea star. The Sunflower Star Lab is based in Monterey. We have a proof of concept lab, and we are building out into a larger facility in this next year. So if you want to help, you can scan the QR code and find more about what we do and where we are on our website. The Humboldt Sea Stars is a new social media platform from Cal Poly Humboldt, which is one of our California partners, and they are looking for sea stars. And again, we have a whole California specific recovery group now working, because there are restrictions between different states on moving animals and the biosecurity from moving animals that we don't know whether they're sea star wasting carriers and they're symptomatic or asymptomatic. So California now has their own little working group, and Cal Poly Humboldt is one of them. Reef Check now is in Oregon and Washington, and if you are a scuba diver and want to participate in community science in your marine environment, check them out. And if you have any other questions, I guess now's the time. Really, thanks so much for everyone who showed up today. And if you want to read more about the sea star wasting syndrome, it's covered in Drew Harvell's book that's meant for the general public. I've read it. It's really fascinating. It talks about not just sea star wasting, but coral disease and other marine invertebrate diseases. I promise you it's not a super downer. It's really fascinating, and they're really trying to find solutions and of these animals that have been around for hundreds of millions of years. And you can go onto the Nature Conservancy's website and read the Roadmap to Recovery if you so choose. And for all the photographers that provided photos, such as this one, this is actually a sunflower star up in Northwest Washington. I can stop my screen sharing.
Awesome.
Keep it. I don't know.
You can leave it on there. That's a nice thing to look at. I was just grabbing the Roadmap to Recovery link and dropping it in the chat. So give me 1 second. There we go. Okay. Awesome. Thank you so much. I have a little soft spot in my heart for sunflower sea stars because I almost lost my hand to one. Maybe exaggerating a little bit, but when I used to volunteer at the Monterey Bay Aquarium, it was half the body was at the surface of the water, and I was feeding it a shrimp or a clam or something, and then it grabbed onto my hand, and I, for probably like 15 seconds, thought I was going to need someone to help me because I couldn't. Their suction is so strong. Luckily, I got my hand away without ruining any tube feet, but I can understand why sea urchins flee the scene when one is coming. Okay, I do have a couple questions for you. Is there a particular age or size when the sunflower star starts to reproduce?
Yes. So this is actually just discovered or proven. Jason, again, at the University of Washington, because he's been doing this research for 4 years and has his oldest cohorts from 2019. He was able to complete the life cycle whereby the sea stars that he grew up in 2019, he was able to spawn and reproduce this year. So usually with many species, it can be age and size. It was about 15 centimeters. So like a normal dish, yeah, size. So not big. They can get 2 feet, 2 to 3 feet when they're really big, but they become sexually mature around 15 centimeters. And I know I'm mixing feet and centimeters just to keep you on your toes.
Great, thank you. So you did touch upon that we're starting to see some sunflower sea stars on the Oregon coast. In June, the Oregon Coast Aquarium found about 25, right, in Yaquina Bay. Since they were completely 100% extirpated, where do you think these little babies came from?
That is a great question. So if you recall, my slide with the, here, let's go back. With the life cycle, they have so they live, I'm going to be obnoxious and just scroll through the whole thing. They live for 50 to 100 plus days in this brachylaria phase. And so they're floating in the plankton for almost 2 months or more, coming from Washington or Alaska, or who knows, depending on, you know, the currents, the Pacific gyre. You're just getting the reason why everyone wants to sail down from BC to California and not sail up is because the predominant winds and currents are from north to south. So we think also from the genetic side, Lauren Scheibelhut, it was able to take historic samples from a decent spread, not a large amount of historic samples, and showed that the population was very well mixed. The only areas where they saw any genetic differentiation was in the really deep fjords in BC and northern Washington, where they might have some isolated populations. But if you're on the coast, I mean, they're just all getting swept down for several days and weeks, and they can just end up there. Or there could be some small enclave of adult Pycnopodia that have survived and weathered this. And that's why we want to be able to create this network of people. I've been calling it like the bat signal starting to work, because, you know, when people see sea stars, again, they're a very mobile invertebrate. So you can't just, you know, say, I found one, let's go back and identify it like you can with white abalone or coral. They usually just stay in the same place, and you can put a little float and come back to it later and everything's fine. But we really need to have a network of people up and down the coast that are trained and on the permit to like collect samples, so we can understand the genetics of where they're from. Like, do you actually see any local or distinct groups, or can we link it? So again, from 1 or 2 samples, we're not going to be able to make grand hypotheses or solve any of the hypotheses, but we'll be able to start building pictures of like, oh, where have these come from, if we start seeing more recruitment or repeated recruitment over time.
Yeah, that was my educated guess. So I'm glad that's what you said.
Yeah, and they do. I mean, this whole like from once they have a gut, the little M in the life history, that's their gut, and they can start eating food. So they eat microalgae, and as long as, again, the thing is, you know, the longer you're in plankton, you're food for something else. So it's really a gamut of luck for these guys to last as long as they do and to settle. So, you know, the further south they have to go, or the longer they have to travel, the less likely their food's going to be hanging around with them, and they're going to find a place to settle. You know, there's a lot going on into that. And one of the cues is coralline algae, which is why you see the picture, which is actually a major cue for a lot of marine invertebrates to settle on. But also a really, really strong response is seen if there's actually biofilm of Pycnopodia, like fresh biofilm. So basically, if they can smell Pycnopodia, they'll be like, oh, sweet, this is a place where I should live, and they'll come and settle. So if there are no Pycnopodia, like they're functionally extinct, then they won't get that strong cue, and they won't get a good recruitment to that area. Yeah.
Very interesting. Lisa has a question. Has the sea star wasting syndrome been limited to the wild?
No. So, well, so yes and no. So sea stars used to be in public aquariums up and down the coast, and now they've sort of dwindled to the facilities that still have them. But what was interesting is that, again, because we take animal welfare very important when we are caring for animals in public aquariums, when the wasting syndrome happened, a lot of the aquariums along the West Coast have flow-through systems. So they pull nice, nutrient-rich, generally, it's a good thing to be able to just pull water from the native environment to bathe all of your exhibit animals in. But in this case, it was not. It was, wasting was definitely spread from the water. And a lot of facilities lost not just sunflower sea stars, but almost their entire sea star population. So that triggered a lot of research in terms of baths with different antibacterial or antifungal or anything. And that's been presented at, that was actually by 2015, the Point Defiance Zoo and Aquarium had shared their wasting treatments that, depending on when you catch it and how early you can actually reverse it. So again, there's a lot of, again, we don't know why some of these things worked, or whether the wasting that we saw is this big wasting syndrome that's still if it's still the same thing that it was 10 years ago, we don't know, because we don't know exactly whether it's a virus and if it's evolved, or if it's, there's lots of things that we don't know yet, unfortunately. But yes, they did see it in aquariums. So yeah, but the aquariums that have them still, you'd think that Birch Aquarium, they never lost their sea stars to wasting. They're the furthest south that have sea stars, and they're warmer waters than they pull from La Jolla. So there's really interesting, why did they survive? Are they is there something more heat? Are they more heat tolerant? I don't know, but now we have at least a genetic full genome analysis of these that have survived. Aquarium of the Pacific doesn't pull in straight from the ocean. We actually have it trucked in by Catalina Water Supply. So they take water from way out in the Catalina Bight. They clean it or filter it, and then twice a week pump it into the reservoirs at the Aquarium of the Pacific, and then they further filter it and sterilize it. So there are many different institutions with different life support that they've seen. Actually, they saw wasting at AOP right in 2013, before it was a big event in Southern California. They'd collected sea stars that were asymptomatic, and that affected the system that they put it in. So they had asymptomatic stars that caused a wasting event, but the other animals in all the other exhibits were fine. So, yeah.
Wow. Crazy. I didn't know Long Beach shipped it from all the way near Catalina.
Yeah. Yeah, and actually a fun fact, Catalina Water Supply also supplies Mandalay Bay in Vegas. So it's a weird world when you start thinking where facilities get their water from, aside from making it on site.
Yeah, right. So talking about safe, there is Trish Mace is on the call, who runs the University of Oregon Charleston Marine Life Center off of our southern Oregon coast, and they have one Pycnopodia. So how does she go about getting
Wow.
helping this?
Wait, so yes, well, please reach out to me, at the Oregon Coast Aquarium and OIMB. I mean, there were two actually at OIMB 2 years ago, and they were both female. They were both collected when they were little, and they sadly wasted that summer. So I'm assuming that this is another individual, but I'm really excited to talk about it, and please get in contact with me.
Yeah, I will. I will connect the both of you via email. This is another one, Trish says. So yes, I'll connect you guys tonight so we can expand the network.
Oh my goodness.
That's all the questions we have so far, unless anybody has any that they just thought of. This is super fascinating, and I'm very excited that it's gained a lot of momentum and support over the last couple of years. And I'm very excited, hopefully, to come to Monterey and visit the lab.
Yeah. Yes, you can reach out to the info at sunflowerstarlab.org. And Vince, who's our wonderful lab manager, will be able to set something up.
Wonderful.
Yeah. And if you're ever like at the Aquarium of the Pacific, please, or any of these institutions, yeah, hopefully you should be able to ask a docent or anyone to see some sunflower sea stars.
Yay. Wonderful. Okay, well, we, I think, are ready to conclude. Thank you, Ashley, so much for joining us tonight. This was really wonderful, and also gives us a glimmer of hope for the future and the recovery of this really, really important keystone species in our kelp forests. So is there anything else you want to leave us with, or ready to go get some dinner?
Yeah, all of the above. No, it's just there's always a way for people to participate, and I love the Venn diagram of like, find something you love, find something you're good at, and find something that you can do for any cause, and just do that thing, whether it's, yeah, there's always a way.
Yeah,
Sea
great.
otters and sea sunflower stars will band together.
Yes, to bring back the kelp forest.
Yes. Oh, well, thank you so much for inviting me. And it's been wonderful being here and hearing also about Elakha Alliance.
No problem. All right, we're getting lots of thank yous in the comments. Thank you for joining us, and we'll see you next month for another engaging webinar. Thanks, everyone.